Hello everyone, welcome to Chandu Biology Classes. So this is the sixth lecture of our series in which we were discussing the most important and high yielding topics for CUET-PG as well as CAT-B. So today is our sixth lecture of the series, which is dedicated to C3 cycle. So in the previous lectures, we have came across different kinds of things like different photosynthetic pigments. Then we discussed about light reaction. Then we discussed about photosystem 1, photosystem 2, as well as your cyclic, non-cyclic photophosphorylation. Then in the last lecture, we discussed about dark reaction, where I taught you in case of dark reaction, like the role of Rubisco, okay, and the mechanism behind the activation of Rubisco, how it is getting activated, the reason that it is you primarily causing the carboxylase activity and not the oxygenase activity. So that is another thing that we discussed, and also, we discussed about the importance of the dark reaction, the regulation of dark reaction we discussed. Okay. So in today's lecture, we are focusing on the C3 cycle that is also known as your Kelvin cycle. Okay. So we will be discussing this cycle. Okay. So I am Deepesh Joshi. Okay, I am an integrated PhD scholar at ISC Bangalore, and I have qualified all these examinations. So basically, our focus in these lectures are for CUET-PG and GAT-B. So I will be touching upon all the topics which are important for those exams, and we will be focusing on question practice as well with each of our lecture. I hope you understood the pattern of our teaching in the previous lectures. So without any further delay, let's begin with the lecture. So basically, if we talk about carboxylation, so what is carboxylation? So as the name suggests, carboxylation means that there is addition of something, okay? And carboxy means that is nothing but carbon dioxide, which is being added to some substrate that is nothing but RUBP, ribulose 1,5-bisphosphate, okay? In the presence of Rubisco to give two molecules of 3-phosphoglycerate, okay? So, being a biochemist, I would like to introduce all of you with the structure of these biomolecules, because if you understand the structure, then it is very easy to understand the cycle. Because if you know the structure, you will know the movement of the different carbons, addition of phosphates, and easy to name the compounds. So, we can look into some structures which are important. Like if you talk about RUBP, that is ribulose 1,5-base phosphate, basically this compound, it is a 5-carbon compound, okay? So which looks something like this, okay? So basically this is a 5-carbon compound. If you name it, you start with giving the priority to the functional group as per IUPAC nomenclature, and you will find out that at 1 and the 5th position you are having the phosphate group attached to this 5-carbon backbone. On the second position you are having this keto group, so that is why it is named as ribulose 1,5-bis phosphate, and is the primary CO2 acceptor in case of dark reaction or the C3 cycle. Now we talk about something that is 3-PGA, that is formed after the addition of CO2. 2 is RUBP in the presence of Rubisco enzyme, which act as both carboxygenase as well as oxygenase, but majority, like primarily, it act as a carboxylase. So basically, it carries out this reaction. So basically, in carboxylation, there is a fixation of CO2, and here you are getting this 3-phosphoglycerate, that is the molecule that is being formed. So if you look into this molecule, basically this is nothing but it is a 3-carbon structural moiety which consists of these kinds of groups. Like in the first position you are having this carboxylic acid, then you are having the phosphate in the third. That's why it is known as 3-PGA. Now in once this is synthesized, what happens is in the reduction step, you use ATP, you use kinase enzyme basically. Okay, so there are two kinds of enzyme which I want you to be familiar with. One is kinase. The work of kinase is nothing but to add phosphate group, and there is something that is called as phosphatase. Okay, so phosphatase enzyme, it removes phosphate group. So here kinase is helping in addition of phosphate group to this 3-PGA and convert this into 1,3-BPGA. Okay, so bis 1,3-bis phosphoglycerate, that is nothing but here if I show you this structurally, basically nothing is happening, but you are having this kind of thing. The phosphate is getting attached to the carboxylic acid group, and you are having this uh, at the last position. So basically, now you see it is 1,3-bis phosphoglycerate. So this is something which is formed after the hydrolysis of the ATP to ADP, okay, in the reduction step of the Calvin cycle. And then you're having something that is called as NADPH. So NADPH, it is getting oxidized. Okay, so with the oxidation, it is forming Na plus and it is releasing the hydrogen ions. So those hydrogen ions, they are being accepted to reduce this. So basically, these are redox reactions in which oxidation and reduction happen simultaneously. So if one species is getting oxidized, another one will be reduced. So this will be reduced from acid to aldehyde and it will form 3-phospho glyceraldehyde, that is also known as 3-glyceraldehyde 3-phosphate. Okay, that is also known as glyceraldehyde 3-phosphate, which is nothing but a 3-carbon compound again. So this is also 3 carbon, this is also 3 carbon, this was 5 carbon, this is also 3 carbon. So here there is no big change. You have this aldehyde group at the rest things, they remain the same. You have a phosphate at the third position. So this is again something which is being formed. And then there is isomer of this that is called as dihydroxyacetone phosphate. So if I like show you with the help of isomerase enzyme, it can convert into DHAP, that is dihydroxyacetone phosphate, which is again a 3 carbon compound that is being formed. And the speciality is that it has this ketone group which is present over here. Okay, and then you are having simple things which are there that are present as it is. Okay, so that is how this is being formed over here. So these are the important structures that you should keep in mind for your examination, okay? And then there is a third step, which is nothing but regeneration. So in case of regeneration, this 3-PGAL, okay, that is being converted into RUBP by a series of reactions. So that is how the entire C3 cycle can be divided into three phases. One is carboxylation, second is reduction, third is regeneration. These are the important structures that you should keep in mind. So if we talk about the phases of C3 cycle, briefly we have understood that there is carboxylation, reduction, regeneration. Now what we will be understanding is how many molecules of these carbon dioxide, ATP, NADPH, they are required to like run the cycle. Okay, so let's understand that in detail. So if I tell you, let's consider you have uh, what do you call? You have a necklace which consists of these beads, okay, and you're having 5 beads. So basically, in one necklace, you are having 5 beads like this, okay, one, two, three, four, five. So this is a 5 carbon sugar that is RUBP, the structure we have seen in the previous slide. So if I take 6 molecules of RUBP and I have to add one CO2 to each RUBP, so I will be requiring 6 CO2 molecules to begin with. In the presence of Rubisco enzyme, it will form an intermediate, that is 2-carboxy 3-keto arabinitol 1,5-bisphosphate. Okay, so this is nothing but this is a 6 carbon. What do we call as intermediate? So now since this is 6 carbon and this was RUBP and this red color ball, that is your CO2. So with the addition of CO2, entire molecule is 6 carbon, okay? And what happens is that at the first and the fifth position you have the phosphate group, okay? At the second position you have this carboxy, third you have keto. So what happens with the help of water, there is a hydrolytic reaction and this gets cleaved, and each of this intermediate compound, which is 6 carbon, it give rise to two compounds which are 3 carbon. So basically from 6 molecules you get 12 molecules of 3-phosphoglycerate. Okay, how it is happening? Since one molecule of the intermediate is of 6 carbons, and once it is getting splitted into 3 carbon 3 carbon, it means one molecule is giving rise to two 3 carbon compound, then 6 molecules will give me 12 molecules of this 3-phosphoglycerate. Okay, so here at the third position you are having a phosphate group in each of the two compounds, and this 3-phosphoglycerate with the help of kinase enzyme which adds a phosphate group, and with the hydrolysis of ATP you get this 12pi. So you hydrolyze 12 ATP, you get 12 ADP plus 12pi. These 12pi, they will be added to this 3-PGA and it will lead to the formation of something called as 1,3-BPGA, that is a phosphate added to the carboxylic group. So if you see the structure, it is very easy to learn. And then with the help of this dehydrogenase enzyme, basically you use a 12 NADPH, you oxidize it, the 12 phosphates which you are, what do you say? So with the help of the dehydrogenase enzyme, what you are doing is you are removing the phosphate, okay? And once you remove the 12 phosphates, you oxidize this thing. So overall, this will get reduced, and from carboxylic acid you are moving on to aldehyde. Okay, from carboxylic acid basically you are moving on to aldehyde. So you are forming 12 molecules of 3-phosphoglyceraldehyde, which is also called as 12 molecules of glyceraldehyde 3-phosphate, and with the help of triose isomerase enzyme you are able to convert it into DHAP, that is dihydroxyacetone phosphate, the structure of which I have shown in the previous slide. You can always refer back. Now, why this Calvin cycle is also called as reverse glycolysis. So basically, if you have one molecule of this 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate, add one molecule of dihydroxyacetone phosphate with the help of triose aldolase. So basically, aldolase is nothing but a kind of lyase which helps in making of carbon-carbon bonds. Okay, so with the help of this aldolase, so since this was 3 carbon, this was 3 carbon, overall you can form a 6 carbon compound. Okay, so the 6 carbon compound is nothing but it is fructose 1,6 bisphosphate. So if you consider this, you will have a phosphate group here, then you will have a keto group, then you will have this HOH, then OH, H, OH, H, and you will have your CH2OP, because you are having two phosphates. One is at the first position, another is at the sixth position. Okay, so this is how this is being formed, and then you remove a phosphate from it with the help of phosphatase enzyme whose work is to remove phosphate. Then you get fructose 6 phosphate, then you use isomerase enzyme whose key function is to isomerize this and form glucose 6 phosphate. So in fructose 6 phosphate you remove this phosphate. In glucose 6 phosphate, instead of this you are having a aldehyde group. Okay, and then you use another phosphatase to remove the phosphate, and finally you form the glucose. Now if you consider the entire process that I have written over here of regeneration of this RUBP, so here we have seen something which we can say as a carboxylation and the reduction, okay, till the formation of DHAP. Now, if you see over here, if you have two molecules of 3-PGAL or two molecules of glyceraldehyde 3-phosphate and two molecules of DHAP, with the help of triose aldolase, we were able to form two molecules of fructose 1,6-bisphosphate. With the help of phosphatase, we were removing this phosphate group and we were able to form two molecules of fructose 6-phosphate, which is a 6 carbon compound. Then in the entire solution pool, you are having these two molecules of 3-phosphoglyceraldehyde. So that is a 3 carbon compound. So now with the help of transketolase, there is a, you can say, transfer of this C1C2, okay, from the fructose 6 phosphate to the 3-PGAL, and basically you are forming the thing but your erythrose 4-phosphate, okay, two molecules of this 4 carbon compound, okay, and as well as your xylulose 5-phosphate, okay, which is a 5 carbon compound. So how is this happening, guys? See, here you are having 6 carbons, so you give 2 carbons to this. So this will have 4 carbon which will be remaining. That is nothing but your erythrose 4-phosphate, and this will become 5 carbon. That is your xylulose 5-phosphate. Two molecules of that and two molecules of erythrose. Now, with the help of two molecules of DHAP, with the help of aldolase enzyme, this will get combined and it will form a pseudohept 1,7-bisphosphate and the pseudoheptulose 1,7-bisphosphate. Again, there will be two molecules, okay, and this is a 7 carbon compound, guys, okay. So this 4 and 3 carbon, these are being combined with the help of aldolase. Okay, this is a lyase category of enzyme. It will make carbon-carbon bonds. Then with the help of phosphatase, you are removing two uh, phosphates. And once you remove them, you are having pseudoheptulose 7-phosphate, which is a 7 carbon compound plus two molecules of 3-phosphoglyceraldehyde. Again, with the help of this transketolase enzyme, what will happen is, since this is a 3 carbon thing, this is a 7 carbon thing, you are giving 2 carbons here, and this will now remain to 5 carbon. That is nothing but 5 mol, that is nothing but your this ribulose 5-phosphate, that is RUMP, because it is a monophosphate, okay, this will be formed. And what you say is that this thing is also formed with the help of this ribose. Okay, so the ribose 5-phosphate, which is again a 5 carbon thing, with the help of the isomerase, it can convert to ribulose 5-phosphate, that is ribulose monophosphate. And then there is a kinase enzyme which utilizes ATP hydrolysis and adds 6 ATP. So this is 6 moles of this ribulose 5-phosphate, and to the 6 moles you are adding 6 phosphates to make 6 molecules of this RUBP, that is ribulose 1,5-bisphosphate. So that means that at the end you are regenerating. So this is nothing but this is a regeneration of RUBP. Finally, you are getting this, and this xylulose 5-phosphate, with the help of epimerase enzyme, it can convert into, so this is a 5 carbon thing and it can convert into your ribulose 5-phosphate with the help of epimerase enzyme. And this ribulose 5-phosphate was again also 5 carbon. With the help of kinase, it uh, gets phosphate group, one extra phosphate, so it becomes biphosphate. And the ribose 5-phosphate, it can also isomerize into this particular compound. So this is the step of for the regeneration of RUBP. If this part is clear to you guys, you can see a very important table that represents the ATP consumption. So we have seen that we begin with 6 molecules of RUBP. We utilize 6 molecules of carbon dioxide initially, okay, for our addition. And also What we saw over here is, as you can see, basically we hydrolyzed 12 ATPs over here. So, and we use 6 ATPs in the regeneration. So in the reduction as well as in the regeneration, we are using ATPs. So 12 ATPs in the reduction phase, 6 ATPs in the regeneration phase, total 18 ATP you have to spend. So this is your expenditure. This is something you are giving. This is something that you have to pay, okay, for carrying or running this cycle. So once you get this 18 ATP, what happens is, so for NADPH, if you want to see in the reduction phase only, this 12 NADPH, as you can see, they get oxidized into 12 NADP, and then you have this 12 phosphate, which will be forming this 3-phosphoglyceraldehyde. So this 12 phosphate will be removed, and then it will be formed with the help of dehydrogenase. So 12 NADPH, they are being again also used or spent. So basically, you are having 18 ATP, you are having 12 NADPH, which are being utilized in this whole cycle. Now, if we understand the energetics or the uh, what you call as the dynamics of the reaction. So, in the very first lecture, I told you that 6CO2 plus 12H2O in the presence of sunlight, chlorophyll give rise to C6H12O6, which is the food of the plant, 6H2O, and there is an evolution of this oxygen. Okay, so that is something which is happening. Now, what you can understand is that to fix 6 carbon dioxide, you are using 18 ATP and 12 NADPH. And then to fix 3 carbon dioxide molecules, you are using how much you have to use? So for 6 CO2, you are using 18 ATP, 12 NADPH. For 3, that is the half of this entire stoichiometry. So you will be using 9 ATP and 6 NADPH for fixing one CO2 molecule. This is very, very important. This is a question that can be asked. You need to use 3 ATPs and 2 NADPH. And we have understood that in the light reaction, so this is about the dark reaction. And we have understood this is photosynthesis equation. This is about dark reaction. In the light reaction, we have understood that by photolysis of two water molecules, you get 3 ATP and 2 NADPH. So that is something being utilized in the dark reaction. That's why we say that light reaction and dark reaction, they are interdependent on each other. Interdependent on each other. So this is something that we are studying from a long time. Okay. So now to use this, like we use 8 photons energy to basically carry out this photolysis. Okay. So we use this 8 photons energy to carry out the photolysis of the what you call as the water. So basically, 8 photons are required to drive this whole light reaction as well as the dark reaction. So this is in terms of quanta, you should know. In terms of quantum yield, you can say, or in terms of quanta, you should understand that to fix one CO2, you require how much? You require 8 photons. Okay. This is something that we can indirectly conclude from the whole photosynthesis. Okay, so I hope this much is clear. Now we can solve some questions. Okay, so basically the first question is in your screen. So you can see that Kelvin-Benson cycle is divided into three phases, namely carboxylation, reduction, and regeneration. The following statements are related to three phases. So product of light reaction ATP and NADPH is utilized in the carboxylation phase. Okay, so what do you think? Is this correct or not? So this is incorrect. They are being used in the reduction phase and not the carboxylation. 6 molecules of 3-phosphoglycerate is converted into 6 molecules of glyceraldehyde 3-phosphate in the reduction phase. That is absolutely correct. Action of aldolase enzyme for the production of fructose versus bisphosphorus takes place in the uh, reduction phase. Okay, so what do you think? Is this correct or is this incorrect? So we have seen that this aldolase enzyme, that is a hydrolase category of enzyme, it is able to produce your fructose. Like basically what happens is that it happens in the regeneration phase and not in the reduction phase. That is something that you have to understand. So this is wrong. Formation of seven carbon compound pseudoheptulose takes place in the regeneration phase. That is absolutely correct. So B and D. So second is the correct option. Moving over to the next question, guys. A student listed following combinations of enzymes and their involvement in different phases of the Kelvin cycle or the C3 cycle. So we have to tell which one of the following combination is basically correct. Okay. So if you see phosphoglycerate kinase in the reduction phase. Okay. So basically, if you remember, I told you that there was a kinase enzyme which was present in the reduction phase, which was helping in transferring the phosphate from the ATP to the 3-PGA and converting into 1,3-BPGA. So basically, that is absolutely correct. Glyceraldehyde 3-phosphate dehydrogenase that is being used in the regeneration phase. So basically, this enzyme that is the dehydrogenase, okay, that is using this in the reduction phase and not the regeneration phase. That is something that you should know that the glyceraldehyde 3-phosphate dehydrogenase, this was the enzyme which was reducing. So I told you there is a dehydrogenase that is coming. So the dehydrogenase, it reduces the 1,3-BPGA basically, okay, and basically it reduces this, okay, to the glyceraldehyde 3-phosphate, and all of this is happening in the reduction phase and not the regeneration phase. So this is wrong. Triose phosphate isomerase in the reduction phase. Triose phosphate isomerase, guys. Okay. So does it comes in the reduction phase? No, this doesn't comes in the reduction phase. Then phosphoribofluorokinase in the regeneration phase. Okay, so in the regeneration phase also you have this 6 ATP which are being converted into 6 ADP. So here also you require a kinase. That kinase was this phosphoribofluorokinase. That was absolutely correct. Okay, so this is something that you should know, and your answer is A and D only. Moving on to the next question, how many ATP and NADPH molecules are required for the synthesis of one molecule of glucose via the C3 cycle? Answer is 18 ATP and 12 NADPH, guys. I told you just now with the energetics. Which enzyme catalyzes the fixation of CO2 in the C3 cycle? Answer is Rubisco, without any second thought. NADPH is consumed only during the reduction phase of the C3 cycle. Absolutely correct. NADPH provides reducing power for the conversion of 3-PGA to G3P, that is glyceraldehyde 3-phosphate. So it provides reducing power because itself it is getting oxidized. It is reducing the another compound that is in the reaction coupled to it. So this is a reduction that is happening, and this correctly explains that why it is consumed in the reduction phase of the C3 cycle and why that phase is called as the reduction phase. So yes, both assertion and reason are true, and R explains A. Match the following enzyme with the role in the Calvin cycle. Aldolase. Basically, I told you aldolase is a lyase which is involved in the carbon-carbon bond formation. Transketolase, it helps in the sugar rearrangement. It transfers carbons, okay, and it forms different sugars. Rubisco helps in carbon fixation. It adds the carbon dioxide to the RUBP. So correct option is the A option. Okay. If six molecules of CO2 enter the Kelvin cycle, how many molecules of RUBP are regenerated? So we have seen that if 6 CO2 you are using, you are generating 6 molecules of RUBP. Okay. So the answer will be 6 without any second thought. So these are some of the questions which you have to solve for the C3 cycle. I hope the lecture is clear to you. In this short time span, I have explained everything in detail that is required for CUET, PG, and GAT-B. Okay, so if you have any doubts, you can post in the comments section. Okay, so let's meet in the next lecture. Thank you for watching. Stay tuned for the amazing content on Chandu Biology classes.